Hello everyone, welcome to today's class. So this is a continuation for the previous class. So what we are discussing is DNA as a genetic material, and where we are discussing about the three major experiments. So those three major one major experiment we already finished, and I shared the link, that is Griffith transformation experiment. Today, I am going to discuss about the second experiment, that is Harvey, McLeod and McCarthy experiment. Right? So, the Harvey and these McLeod and McCarthy, these are the colleagues for the Harvey, and they have done a little more extension study on the Griffith experiment. So, here we should understand that Griffith experiment. And Griffith experiment has proven that there is a transforming principle, that is transformed from the heat killed S strain to the R strain. But he has not given any information about what is that transforming material. Now, that transforming material they want to prove, so that these three scientists, that is Harvey, McLeod, McCarthy, has taken an extension work of Griffith experiment, and they did a beautiful experiment which primarily confirmed that DNA is a genetic material, not RNA and protein. So, if you see the basic details of this experiment, right? So here, this is like what we are saying, which is to be on a little extension, or which is like an extension work of Griffith experiment. So here, this is all they done in the bacteria, that is Diplococcus. But the Griffith experiment did in bacteria, that is Streptococcus pneumoniae, that we should understand. The Streptococcus pneumoniae is a bacteria. It can be used for transforming experiment by the Griffith. But in the Harvey, McLeod, McCarthy experiment, where they used Diplococcus bacteria. So here the principle is same. First, this experiment they done in the test tube, not in the laboratory. So we call them as, which is an in vitro, not in vivo. So what do you mean by in vivo? If you see the Griffith experiment, that experiment we call it as in vivo experiment. Why? Because where the live mouse is used for the experiment, but here no organism is used, like which is not infected with the mouse, which is only done with the test tubes. All right, this is all related with the what the material, the notes which are how they proceeded for the experiment. But anyway, this thing you can anyway better understand when we go into the is actual experiment part. So now see, this is what exactly major idea of their experiment. So if you understand this one, then you can better understand the previous one. Okay, then if you see this, see, this is like a very simple experiment. So initially they have taken a strain that is called eat kill S strain, and we also I am forgetted to tell you in a previous experiment, this S strain is having the number that is, we'll call it as 3S strain, right? Then R strain will call it as 2R strain. That is like in a 3, 2, which are related with this strain number, right? You temporarily you just forget it, this 3, 2, but the major concept is here the eat kill S strain they have taken. It's clear. So this eat kill estrange solution, first it can be divided into three portions. This is 1, this is 2, and this is 3. This is important. So whatever the solution or whatever the culture that is present in the this test tube, this we'll call it as master test tube. In this master test tube, whatever the solution that is there, that is related with the heat kill strain. So this heat kill strain culture is divided into three different test tubes, 1, 2, 3. And in the first test tube they added into an enzyme that is called proteinase. Clear? Then on second test tube they added the enzyme that is called RNase. And in the third test tube they added DNase. Right? This we should understand. What is the function of proteinase, what is the function of RNase and DNase. So this is very simple. For suppose this heat kill solution, that heat kill strain bacteria, if you add the proteinase, what will happen? The proteinase is an enzyme that can digest all the proteins. Digest all the proteins mean, what does it mean? There is no enzyme or there is no protein which is in a functional mode after adding this. అంటే ఏంటి? ఇది సింపుల్. ద ప్రోటీనేజ్ అనే ఎంజైమ్ను యాడ్ చేసినప్పుడు ఏదైతే హీట్ కిల్ ఎస్ట్రెయిన్ బ్యాక్టీరియాలో ప్రోటీన్స్ ఉంటాయో అన్ని ప్రోటీన్స్ అన్ని డిగ్రేడ్ అయిపోతాయి. సో డిగ్రేడెడ్ మీన్స్ నాన్ ఫంక్షనల్. క్లియర్? సో వెరాజ్ ఇఫ్ యూ టేక్ సెకండ్ టెస్ట్ ట్యూబ్ RNase, the name itself indicates which can degrade RNA. So RNA ను బ్రేక్ చేస్తాయి. Right? If you take the third test tube, the DNase mean which can degrade the DNA. These are all three enzymes. Anyway, this we should better understand when we are going into the recombinant DNA technology. So the proteinase all destroy the proteins, RNase will destroy the RNA, DNase is destroy the DNA. Is clear? But where it is to be happening? In the heat kill strain. Right. So at the end of that, no protein because the proteinase K, no RNA in this sample because the RNA is degraded, no DNA in this 3 test tube because the DNA is degraded. It's clear? Now, taking each and every this test tube, one test tube is number 1 is added into the conical flask where he has taken the live R cells. So far from this part, this is related with the heat kill strain. Now this one test tube solution is added into the conical flask where R cells, live R cells are present. So I think you might have understand about S and R cell. S cell is the one which causes the pneumonia. R cell is the one which is not causing the pneumonia. Right? So anyway, your heat kill mean what is happening? The even S cell is causing the pneumonia, but if you are eating that particular cell, definitely this will kill. Kill mean there is no function of the this S cell, so there is no pneumonia. So you are adding this content into the this conical flask where live R cells are present. Right? Then they observe that S cells appear. It's clear? S cells appear. So what do you mean by S cells appear? That we will see. All right. Then if you take the RNA, the second one is again added into the second conical flask. Right. So here also the S cell appear. S cell appear mean, what does it mean? The heat kill cell is present here in this. And if you take the third one, this is DNA, where if you add the R cell with this solution, there is no S cell is appear. What does it mean? This is very important because here the HOR cell, there is no transforming principle to the S, so definitely SL is appeared. There is no transforming principle to the again the second, so definitely here also the SLs are appear. The third one is here the DNA is transforming into the R cells. This is important. The whatever the DNA that is present in the heat kill SL that is transformed to the R cell. So this is not appeared here. And if you take these R cell, definitely this R cell will cause the pneumonia. Initially, R cell will not act as an pneumonia causative agent, but after addition of this solution, then they found it that can acquire the DNA of the heat kill S cell, and it is converted into the R cell is converted into the S is converted into the pathogenic, so it can cause the pneumonia. That is with the help of it is taking the DNA from the S cell. So this confirmation, this is the first confirmation which I have given that transformation requires DNA. So therefore it is a genetic material of this cell. So here no protein and no RNA, only DNA will acting as a transformation, so that this first experiment, initially the Griffith has proved something which is transferring. So he has given transforming principle, but he has not given which is transforming, whether protein, whether RNA, whether DNA. But the second experiment, this is RV experiment, they first confirmed that this is no protein, no RNA, only DNA is transformed. So that's what I have written in the previous slide. So this is the major breakthrough to the confirming of DNA as a genetic material. So here one more thing which I am explaining in a very simple manner, you just understand this one about the RV experiment. But suppose if you take the A is an strain, R strain when you culture in it, it can give you the rough colonies. If you take B, where this is strain S, this is heat kill strain. So if you culture, after heat killing, if you culture, there is no colony. What does it mean? The heat kill strain is destroyed. So there is no colony formation. Colony formation means number of colonies, number of divisions the cell can go. Then if you take the C, the DNA isolated from the this S cell, then culture, no colony. Even if you purely isolate the DNA and you culture it, no colonies is formed. Right? It's clear. And if you take the D, so live R cell and heat killed S cell, if you mix these things and if you culture it, we will find the colonies, right? Then SL colonies. What does it mean? So the transforming principle is happening. So the same way, which is same, like anyway this document I can send it to you, because at finally we should understand that the H you can see here. So here this is live R cell and DNA. So this is, if you treated with the DNAs, what does it happening? It cannot form the colonies. What does it mean? So here the transforming principle is DNA. But if you treated with the protease, the colonies are formed because the protein is degraded. Even though the colonies are formed, mean that is the transforming principle is not protein. Even though transforming principle is not RNA, even though the protein, the colonies are formed, mean the transforming principle is not the RNA, not the protein. This is how the experiment has proved, and this is the first experiment which shows the DNA will act as an genetic material.